We are going to talk about a tool called rROMA, which is the R version of the ROMA tool. You can see the publication here that can be used to functionally analyze your expression data. So it can work on bulk RNA-seq data. It can also work on single-cell RNA-seq data. So it was implemented by the Marie Curie Institute in France. So you can download and install it from their GitHub page. It uses a number of additional packages like Skater, Biomart, and ggplot2. So it will only work if these three packages have also been installed on your computer. So in the accompanying tutorial, you can find the code for installing these three packages and for installing the rROMA package itself. You have to load the packages before you can use them. So we are going to start with bulk RNA-seq data. So I will load the DE-seq2 package. Then I'm going to load a number of additional packages. Okay, so this is the package for the functional analysis. This is a package to get the bulk RNA-seq data in the correct format for rROMA. These two packages allow us to convert ensemble IDs because in our bulk RNA-seq data, all the genes are represented by ensemble IDs. We are going to convert them into gene symbols, and for that we need these two packages. And then here you have an additional three packages that are helpful for creating plots of the results. So rROMA can be used to do downstream functional analysis of RNA-seq data. What do I mean with downstream? I mean that typically you will first analyze the data, you will find differentially expressed genes, and once you have done that, you typically want to know what pathways are affected by the experiment that you have done. And this is where rROMA can help you. It's a tool that is similar to gene set enrichment analysis tools, or tools that perform what is called functional overrepresentation analysis like gProfiler or David, but it uses a different approach to find relevant pathways that are affected in your RNA-seq data. We're going to load the data. We're going to start with bulk RNA-seq data. So the input data needs to be processed. So rROMA cannot work with raw counts. The counts need to be either variance stabilized and normalized values when the data come from a DE-seq2 analysis, or CPM values when the data come from an HR analysis. So DE-seq2 and HR are the two main packages that you use to find differentially expressed genes in your bulk RNA-seq data. So depending which of these two packages you use, you either convert your data into VST values or into CPM values. And these transformed values are the ones that you need to use for rROMA analysis. So I'm going to set my working directory to point to the folder that contains the count files. So we need to load the sample information. So the sample information is just the names of the samples, the names of the count files. You have typically one count file per sample. And then additionally, you see here the grouping information of the samples. So this sample, for instance, the 09 sample is a treated sample and comes from the cell line with this name. So the grouping information divides the samples into different groups. You need to tell him the name of the folder that contains these count files. So in my working directory, there is a folder called htsec counts, and this folder contains the count files. I can show you. So here you see it. This is my working directory. There is a folder here that's called htsec counts, and this folder contains the count files. I import the data into what is called the DESeq dataset object. I specify the group of samples that I want to use as a reference. So in this case, I'm going to use the control samples as a reference. I remove all the genes that show almost no expression. So all the genes with a total count over all the samples that is lower than 10 will be removed from the dataset. And then I analyze the data. I'm going to compare the treated samples to the control samples, and I'm going to do this comparison over all the cell lines that have been used in the experiment. So you see that he is here normalizing the data. That's what this line means. So he normalizes the data to make the samples comparable. After this normalization, all the samples will have the same total count. He will estimate the variability for each gene in each group of samples, and he will try to make the variability more accurate, because as you can see, we only have 4 replicates per group. That's not enough to get a good estimation of the variability within the group. These 4 steps are all linked to making the variability more accurate. And then this last step here, fitting model and testing, well, this is the step where he's going to do the actual comparison. So for every gene, he will compare the counts between the treated samples and the control samples, and he will check if there is any evidence that the gene is differentially expressed. So in this DESeq dataset, you see that every gene is represented by an ensemble ID. This is not ideal because later on in the rROMA analysis, we are going to use gene sets, so groups of genes that have functionally something in common, for instance, genes that belong to the same pathway. And in these gene sets, you will see that the genes are represented by gene symbols and not by ensemble IDs. Now, in the rROMA analysis, you always have to make sure that there is a match between what is used to represent the genes in the gene sets and what is used to represent the genes in your data. So this is not the case here. So we are going to convert these ensemble IDs into gene symbols. To do this, we are going to use the human annotation. So in these 2 steps, I'm retrieving a table that will contain, for every gene in the human genome, the mapping between the ensemble ID and the corresponding gene symbol. Then I convert the ensemble IDs from my dataset into a data frame. So this becomes a data frame with just one column, all the ensemble IDs in my dataset. And then I join the 2 tables based on the column called ensemble. So you see that that data frame that I created here with the ensemble IDs from my bulk RNA-seq dataset has a column called ensemble with ensemble IDs. And as you can see here, that table S that I created that contains ensemble ID and corresponding gene symbol for all the genes in the human genome also has a column called ensemble with ensemble IDs. So based on this column called ensemble, I can merge the two tables. And that's what I do here. Some ensemble IDs will be linked to more than one gene symbol. It doesn't occur very often, but it happens. So we will have duplicates in the resulting merged table. So that will be an issue in the downstream analysis. So I get rid of these duplicates by running this code here. So for every ensemble ID here in my merged table, I'm going to check whether it's duplicated, yes or no. And if it's duplicated, I'm going to remove it. I have normalized my counts. This is what I have done here in this step, so with the DE-seq function. But I haven't yet variance stabilized them. So I did get rid of the differences between the different samples, but I did not yet get rid of the heteroskedasticity issue. So to get rid of the heteroskedasticity, I perform this transformation on my counts. So I have 20,000 genes now. Initially, I had a lot more genes, but I performed this step here. Remember, I removed all the genes with very low expression levels. And as a result, I have 20,000 genes left in my data set, and I have 8 samples. So at this point, I have the data in the correct format, but the data is still stored in a DE-seq dataset, and rROMA cannot use a DE-seq dataset as input. It wants a regular count matrix as an input. So from this DE-seq dataset here, from this one, the one with the variance stabilized and normalized counts, I am retrieving the count matrix using the assay function. So this will generate, as you can see, a regular matrix with my counts. So in this matrix, I'm going to use gene symbols as row names. So not ensemble IDs, but gene symbols to match the gene sets that I'm going to use later. So that's what I do here. I define the row names of this matrix as the column called symbol that contains the gene symbols in my data frame that I created here. Now, some of these gene symbols are missing. Not every gene in human has a gene symbol. I will scroll down. You can see that here. Many of these gene symbols are missing. This will cause issues later on in the analysis. The gene sets, they will contain only genes with known gene symbols. So that means that these genes here will not contribute whatsoever to the rROMA analysis. They will not match any genes in the gene set. They will only cause issues in the analysis. So I'm going to remove them. So this is what I do here. I check for gene symbols. They are used as row names now, remember. I check for gene symbols that are equal to missing values, and I only keep the ones that are not missing. As a result, the matrix becomes a bit smaller. So now I really have the input for the rROMA analysis, a matrix with gene symbols and corresponding variance stabilized and normalized counts. For the rROMA analysis itself, this is enough, but to check the rROMA results, we also need to have the sample annotation. So we need to know which sample belongs to which group. So we have that information, it's this information here. And unfortunately, in rROMA, you can only specify one grouping variable. So we are going to focus on the one that is important to us here, and that's the treatment. That's the main thing that we want to do on this data set. We want to compare the treated samples to the control samples. So the sample information needs to be in a vector. So this will look in the DE-seq dataset in the call data and retrieve the column called treatment, which will be the same column as the one that you see here. So it will produce, as you can see here, a factor with two levels, either control or treatment. Now this needs to be a named vector or named factor. So that means that you have to give every element of this factor a name. How can we do this? Well, the names should be the names of the samples. What are the names of the samples? The names of the samples are used as column names in your DE-seq dataset. So these sample names are going to be used as names for the vector that contains the grouping information. So when we take a look at groups now, so you see that every element has a corresponding name.